CME credit, used a cell phone, screen back in the back there with a 2D barcode. And if you are not a CHOA physician, they have certificates in the back. Please see Aaron back in the back corner if you want to take care of that. All right, so Dr. Whiteley, Dr. Marvin Whiteley, grew up in Texas, and that'll be very apparent as soon as he stands up and walks over here and you hear his voice. That's not a bad thing. I lived in Texas for quite a while, too. Marvin earned a Bachelor of Science in Zoology at UT Austin, then a Master of Science at Texas State University, where he first started working on bacteria and biofilm development, and that's been his bag since then. He did a PhD at the University of Iowa, where he studied quorum sensing and biofilms in Pseudomonas aeruginosa under the direction of Pete Greenberg, who's considered the father of the field of quorum sensing in Pseudomonas, which is, of course, one of the most important pathogens in CF. He did a brief postdoc at Stanford before launching his independent career as faculty in Oklahoma, at the University of Oklahoma, then moved back to UT Austin, where he rose through the ranks and became professor and director of the Center for Infectious Disease there. After I got to know Marvin a bit from serving on a grant review panel with him for a lot of years, and a bunch of his friends that think highly of him are people that I also call friends, so that was a vote in his favor. We were really fortunate to be able to recruit him to Atlanta in 2017, where he's now the Benny and Nelson Abel Chair in Molecular and Cell Biology at Georgia Tech in the School of Biological Sciences, and he's also a GRA Eminent Scholar. And now he's actually co-director of our CF Atlanta Research Center, which you know is heavily Emory and Georgia Tech and Children's working together. And he actually serves as the anchor faculty member there for Georgia Tech. He's helping to draw into the CF effort a bunch of other researchers and trainees on that campus that's, 5 miles and about 2 hours away from us. And he's actually recently gotten funding from the dean there to launch the Center for Microbial Dynamics and Infection that will leverage his connections to CF Atlanta. And of course, CF Atlanta is going to leverage the hell out of that at Georgia Tech. He's an award-winning teacher and researcher, and he's really impacted the growth and quality of our research in our center, particularly in the microbiology domain. And he's been challenging the way we think about microbes in the airways of our patients. And I think you're all going to learn a lot by listening to his presentation today, where he's not going to focus on just one bug at a time, but really think about the complex environment that our CF patients deal with in their lungs. And one last great thing to know about Marvin, related to the fact that he's from Texas, is he's probably the only faculty member I know of in the entire city of Atlanta who wears boots more than I do. Thank you, Marvin.
All right, thanks. So it's good to be here. Can you guys hear me? Is this okay? All right. Yeah, so it's good to be here. I give a lot of talks, and a really diverse audience. I was really sort of going back and forth about what you wanted to hear about. I run a pretty big lab. We work on a lot of different bacteria, but I'm going to focus on only a couple of these bacteria, things I thought you might be more interested in. Usually when I give talks, no matter where it is, there's a question and answer section, and it's me asking you questions, sometimes also. So you're all physicians, right, so you speak up early. So I'm not, and so if I forget something, I may ask you. So my lab works on a lot of different things, and a lot of you I think a lot of people who think about microbiology think about it very simply. You have these little bitty bugs, and they do stuff, and in your case, you think they do bad stuff, right? And then you want to somehow manipulate them so that you get rid of them, or they do good stuff, right? That's kind of what we think about it. It's actually a lot more complicated than that, and those are sort of some of the points that I'm going to try to make today. And actually, studying microbiology there's been a couple of renaissance in microbiology. In the late 1800s, there was this big renaissance in microbiology. Does anybody know why?
Microscopes.
Yeah, well, the microscopes were in the 1600s with Leeuwenhoek. He came about. 1684 was the first time a human ever saw a bacterium, was in the 1600s. What happened in the 1800s? Anyone know late 1800s? You got read the culture. Right? Koch and others made bacterial plates. We could actually see them. Pasteur was growing them in test tubes, isolating them out, Koch's postulates came about, where we figured out that organism X causes disease X, and we started thinking about that. That was a big renaissance up until about the 1900s. The next generation, the next sort of, I think, big move in microbiology was in the 50s, and it was really genetics. It was when we were able to actually go in and manipulate genomes and actually infer functions from manipulation of those genomes. How do you copy your DNA, right? How do you make a person sick and finding toxins? And I think now we're in a new one, and that is with the microbiome and microbiota, and really thinking about things as holistically, which is what we're going to talk about. But this last question is actually a lot harder than knocking out a single gene. It's actually much more difficult. You need to think about communities. So today we're actually going to take parallels from ecology and try to propose that they're useful, and these concepts are important for infection. So we do a lot of things in the lab. You see some images up here. There's in vitro, basically growing in test tubes. Of course, we use mouse models like most microbiologists who are interested in pathogenesis do. But we also now have sort of moved into humans and actually getting human-derived samples to work with. And also, my lab has spent a lot of time developing new technologies. I think there are some big questions in microbiology, and we can't answer them with the techniques that we have now. We need to develop new techniques. So we're going to focus on two things. The first part is going to be looking at transcriptomics. I'm going to tell you what that is in a second. And then the second part is going to be the development of new technologies. So the thing that you need to know when sort of, I guess, one of the first slides I always use is that bacteria are actually social. A lot of people, even today, still think of bacteria, these sort of unicellular entities that usually divide very fast. And then they sort of act as individuals, but it's actually not true. Actually, most bacteria have some sort of sociality that they've evolved, some mechanism for them to actually interact in some ways. I'm going to show you a video here from YouTube. This is not a bacterium that you need to worry about, though, so don't freak out. Your patients aren't going to get this one. This is Myxococcus xanthus. It's this bacterium right here. This is a bacterium that actually lives on dead and decaying logs. And this bacterium, what it eats are other bacteria. And it actually hunts in groups called wolf packs that move and hunt together on these logs. And this is Myxococcus eating an E. coli colony. And so this bacterium is incredibly social. And this is the most socialistic bacteria we have. And the reason is it has developed mechanisms that when they move together, if one tries to divert out, it is ejected from the group and not allowed to participate in the feeding. And so this is a really complex social behavior, but it's not unlike the bacteria that you actually are going to be working with and are seeing in your patients. This is actually up here, which my pointer doesn't seem to be working. This up here is Pseudomonas aeruginosa, a mutant of Pseudomonas aeruginosa that forms these sort of beautiful sort of colonies on plates. There are specific genetic markers that allow mutations that allow us to do it. What you're seeing on the bottom right is a bacterium called Vibrio fischeri. This bacterium actually makes light. It makes blue light. And that light is actually used for this bacterium to interact with animals in the ocean to actually provide them with light so that they can use it for all sorts of behaviors. And all of these social behaviors require communication between the bacteria. They count their numbers, they know who's there, they know which bacteria are around. So today, think about this when we're thinking about the subjects I'm going to be telling you about. All right. Well, that might be the last slide. I don't know what's happening. Oh, okay. The other thing that we have to remember, and hopefully you all, because it's in the name microbiology, is that bacteria are small, right? And I know that you think, oh, well, we all know that bacteria is small, right? But I don't think people really realize this when they think about microbiology. So let's say this is a sample of cystic fibrosis, expectorated sputum. And let's say there's a couple of mils of it there, and there are 10 to the 9th bacteria. So there's 1 billion bacteria per mil. So how big is a bacterium? Does anyone know size-wise? Everybody said micro, right? How big? Are we in the nanometer or a micrometer scale? Micron scale, what's a good number for a bacterium you would treat? 1, that's a good number, right, about 1. There are a lot of bacteria that you see that are associated with the human body are about 1 micron. There are actually bacteria that you can see with the naked eye. Some are almost a millimeter, right, but you're never going to see those in a patient probably. All right, so anybody know what the volume of a bacterium is? In liters? In mils? In any unit, ounces? Whatever you guys want to do. Centiliters. So if you have 10 to the 9th bacteria per mil in here, how much of the volume are these bacteria occupying, do you think? If you look at an infection, what does it look like? Is it like packed with bacteria, do you think? Do you think it's really packed fully with bacteria? So it's not. And in fact, it's really hard to find them when you look. About 10 to the 9th bacteria, and this is a big bacterium that you would think about, is 1 microliter. So that means that over 99% of the volume of that expectorated sputum is host material. There are no bacteria, okay? So when you think about infection, and CF is actually very high levels of bacteria. If you look at tissue infections and those kinds of things, volumetrically, there's much less bacteria than that, okay? So when you're treating an infection, you're not treating this community that looks like a colony on a plate. It's actually very diverse, very sparse sort of landscape. And I think of it more like if you think about the African savanna, right, and you sort of think about zebras around. That's sort of what you should think about. All right, so the thing we're going to talk about, like I said, a lot of my lab works on a lot of bacteria. We're going to talk about 2 today that you guys know about, I'm sure, Pseudomonas aeruginosa and Staph aureus. And the reason is because these are generally and oftentimes found together in infections. They're actually found oftentimes more together than apart when they cause disease. Generally chronic diseases, not necessarily acute. The other thing that you see is that they actually interact with each other, at least in our animal models of infection, and they display something that's called synergy. And so this is a term that's been around in microbiology for about 60 years. And what synergy is that when you put 2 bacteria together that in an infection, they cause a worse infection than each one does alone. And a lot of you probably say, well, all bacteria do that. Actually, synergy is very rare, incredibly rare. People have actually gone through and done pairwise interactions in life to show that rarely do you ever see synergistic interactions, okay? So these are special, these are not normal, okay? These are very prominent in CF, but also in chronic wounds. And we're going to talk about both of these types of infections today. So the question is, how do you study these types of chronic infections? And the question is, microbiologists, since Pasteur and Koch started growing bacteria on agar plates and in test tubes, they use model systems. And so I'm going to go through for about 4 slides, tell you about model systems that are used to study bacteria, how my lab has used them successfully. I'm not going to tell you about the hundreds of unsuccessful things, but I'm going to tell you about the successful ones. And then I'm going to tell you, and then we're going to ask the question, are these models actually useful for understanding the diseases you're trying to treat in a human? Okay, that's all we're going to do today. It's pretty simple. All right, so the first model system, Pasteur was doing this, Koch was doing this. Lots of folks are doing it. You basically grow them in a test tube. This is Pseudomonas aeruginosa growing in a test tube. It forms this beautiful bluish-green color. It's called thiocyanin. And we've used this a lot to actually identify all kinds of things about bacteria. So we've learned how DNA replicates using this system, right? And we didn't do it in eukaryotic cells. We did it in prokaryotic cells, and then applied those principles and asked whether the same similar sort of things happened in eukaryotes. A lot of the biochemistry has been done here. So, but of course, the media that we oftentimes use here are basically the same ones that Pasteur did. So you take some beef and you boil it, and you take it and then you take all the chunks of beef out, and then you use that to grow bacteria. That's still what we're doing. Sometimes we use plants and yeast and other things in these media, but we're still using the same thing. So it's probably not the best thing, but it allows us to do some things. When I was in grad school, we started doing this, and all of you probably, has everyone heard this word? Did they tell you this in med school? I've only taught med school for 1 year before, right? And they all tell you biofilms, right? Is this the picture you see when they show you biofilms? I don't know, these are the famous pictures, these beautiful sort of mushroom pictures, right? This is Pseudomonas aeruginosa, there's a surface here, and this is the bacteria. This is hundreds of microns. This is literally hundreds of bacteria attached. So when I was in grad school, if you didn't work on biofilms, you weren't cool. And if you didn't grow mushroom biofilms, you couldn't publish Nature papers, right? But if you can grow a mushroom biofilm, which took me 6 months when I was a grad student, because these are really hard to grow. Because, in my opinion, they don't exist anywhere on the planet except in a lab, okay? I've never seen them anywhere else, and we've done a lot of imaging. But the thing that it did, made us start thinking about some concepts that microbiologists didn't think about. And the biggest one here is biogeography. We'll get back to that one towards the end. But biogeography is just the study of where things are in space and time, okay? And you think, well, why the hell would that matter for me when I'm treating an infection? I'm going to tell you why at the end. That microbes are just like us. They're biogeographically constrained, and they have to be in certain spots. And if they're in the wrong spot, they don't cause disease. If they're in the right spot, they do. All right, so I'm going to tell you kind of one example just from my lab. This is a grad student from my lab, Astoria, and I'm going to tell you how this is really the only time that I think using an in vitro system, test tube type system, worked in our lab to find something that I think might happen in a human. And what Astoria was really interested in was Pseudomonas, and they made that beautiful pigment called thiocyanin I talked about. So if I would have asked you what the first antibiotic was, what would you have said? And penicillin, right? Yeah, so there were a whole slew of them before penicillin, lots of things before penicillin. Actually, the dye industry was actually one of the most important industries for the first antimicrobials. The late 1800s, everyone started liking dyed clothes of different color. And it actually turns out some chemists went in and took all those dyes and looked for ones that could kill bacteria. So the first antibiotics were actually these red pigments that would actually treat syphilis. The problem with them is they dyed you red when you took them. And this actually became in London, it became a really big deal because people walking around who were dyed red, people knew were sick, right? And they didn't want to be around them because they knew that they were sick. And so there's been a lot of molecules before that. This molecule actually, though, preceded even those. This is the very first molecule that was ever purified and injected into a human to treat a bacterial infection, at least that I've been able to find. And it was used to treat some individuals who had life-threatening lung diseases. And so thiocyanin is an antibiotic. It does kill other bacteria. The only problem with using it as an antibiotic is it also kills humans really, really well. And if you look, they published a paper and half the patients died after they injected them. They had a small sample size, thankfully. But a couple of them did get better, and then they said this is something that could be, this was a good antibiotic. It only killed half the people, and a couple of people got better. And so they actually used this until the early 1900s to treat skin infections in humans. And it was purified from this bacterium. So the nice thing about thiocyanin is it is a virulence factor in Pseudomonas. It helps Pseudomonas make you sick. But the other thing that thiocyanin is it's co-regulated with lots of other things that make you sick. So it can be used as a biomarker. When you see blue, that's virulence. That's what we think, right, in Pseudomonas. So what Astoria did, she had a very simple thing that she did, is she actually took Pseudomonas, put it into a 96-well plate, and isolated a bunch of bacteria from wounds, from human wounds, and mixed them with this, and then asked, do you ever see more thiocyanin, right? That kind of makes sense that, well, if it's more blue, then maybe this other bacterium is promoting Pseudomonas virulence, right? That was the idea. And what she found when she did this is lots of bacteria promote Pseudomonas virulence. And the main ones that did were gram-positive bacteria, all the gram-positive. Does everyone in here know the difference between gram-positive and gram-negative? I'm going to tell you anyway. So these are the two. This was Hans Christian Gram in 1864, actually came with ways to classify bacteria. And he classified them based on how they stained. Okay, but that staining actually inferred some biochemical properties of these two bacteria. So gram-positives are bacteria that have a cell membrane surrounding their cytoplasm, and then they have peptidoglycan around them, about 60 nanometer walls. That's what they have. Okay? And peptidoglycan, all of you probably heard of it, it's a structural component. It contains sugars and amino acids. This is a structural component that kind of holds the bacterium together. Gram-negatives have that same structure. cell membrane peptidoglycan, but they put another membrane on top. This is where LPS is, okay? So that's the difference between them. The problem with gram-positive bacteria is when they actually divide, because that cell wall is on the outside and not contained in another membrane, when they divide, they have to restructure that wall, right, and make it grow. And when they do, they shed components of that wall. It turns out what Pseudomonas is actually sensing in gram-positive bacterium is this peptidoglycan wall. So this bacterium has actually evolved a receptor for a molecule on another bacterium. And when it binds that molecule, it turns up and ramps up its virulence. And so the mechanism that we've seen, that I told you about, synergy, where the infection is worse when Pseudomonas and staph around, is largely mediated by this interaction. Like Pseudomonas sensing a piece of the other bug and then turning on virulence. And this all came about, and so Ashwari actually went in, found the receptor, went into mouse experiments, and showed that this worked. Like I said, this is probably the only time that something from the lab, from a lab medium worked for us. All right. So I like to use analogies. Some of you won't like this, but oh well. I like to think about it like this. If you were studying black bears, which it would be cool to study black bears, I wish I would have studied black bears, and you wanted to know something about black bear biology, right? And I gave you this data, right? This is data, right? Probably better than some of your grad students generate, right? So it's a picture of a bear that invaded a house, and this is a piano. What would you think if you saw this data, right? You would think that black bears, maybe they live in houses, they walk on 2 feet, and they play the piano. I mean, it's a reasonable assumption from this data, right? This is kind of how I think most test tube experiments are, right? Now, if you wanted to feed this bear pizza and then ask how it digests the pizza, that would probably tell you the same as it digested it here or out in the woods, right? The digestion is set. Biochemistry is set. So that's what I think is the same for bacteria. If you want to study DNA replication, a test tube is great. But if you want to study social behaviors, you can get in trouble. So what do you do then if you want to do something better than a test tube? Well, one thing that you can do is, well, maybe I can try to mimic the environment that I'm trying to study, right? Instead of just putting them in some beef broth, right, maybe I'll try to mimic it. This is what we've sort of done with cystic fibrosis. So what we've actually done in cystic fibrosis is we took expectorated samples from patients, went in and did mass spec, and said, what's it made of, right? What chemicals are in it? What's it made of? And then we make it in the lab. We call it synthetic CF sputum medium. And it looks like this. It has the same viscosity, and we've done all kinds of experiments in it. But the question has always been, and of course for reviewers, right, they always look for something to complain about. And so what they're complaining about is, well, how do you know this is good? Just because it nutritionally mimics, right, doesn't mean it's any better than LB or some of these other media that we use. How can you test that? So the way that we actually tested this to ask whether this medium is a good surrogate for the sputum that you get out of a human is we use a genomic approach. You're going to see this approach a couple of times. It's very simple to understand, though. So what you're actually looking at is called transposon sequencing. All of you probably know what a transposon is, but again, I'm going to tell you. Anyway, a transposon is a genetic element. It's a small genetic element that's mobile. It's actually discovered in corn, right? A lot of you know this. These hopping genes, jumping genes you probably heard about. At some point in your educational career, you heard about jumping genes. We use them in bacteria, though, to actually do genetics. This was the revolution that really allowed genetics to happen in bacteria. So what you can do with transposons is you can introduce a single transposon into a bacterium, and it will actually hop randomly onto the chromosome. And when it does, it interrupts wherever it hops, right? If it were to hop in the middle of a gene, it would disrupt that gene. And so what you do is you make hundreds of thousands of these transposon mutants. Each one of these are sort of each one of these colors is a different transposon mutant. So the transposon's interrupting a different thing in the bacteria. And because we can actually, and by using next-gen sequencing, which you don't necessarily need to know, we can actually count the number of mutants here, and then we can put them through some selective condition. And what we did is we put them in sputum from a patient and sputum that we made in the lab. And then we let them grow, and then what we do is we count them again. And what you're actually looking for are mutants that don't do well in these conditions, right? So let's say this pink one, right? It was really highly prevalent in the transposon mutant library. You put it into the sputum, and now you don't see it again. What does that tell you? What does that tell you about where that transposon went? That area is probably important, right? Right? Because now it doesn't grow in sputum. And now you can assess 100,000 mutants simultaneously and all the functions associated with that in one experiment. So we did this, and we just you have to come up with some sort of clever computational methods to analyze this data, but we just did a very simple experiment. What genes do you need to grow here? What genes do you need to grow here? Are they the same or are they different? If they're different, the reviewers are right. If they're the same, then I'm right. This is basically how I looked at it, right? When you actually do this experiment, of the 5,000 genes that we looked at, it was only 14 were different. Okay, and you go, well, is 14 good? Well, if you use those other media that I was telling you about and compare it to what the human CF sputum, it's over 200, probably around 350 or so. So just simply making up a medium that nutritionally mimics what you see in a human gives you some advantages, right? It may allow you, hopefully, and this data would tell you that functionally this medium is going to be more useful for learning what's going on in a human. Okay? All right. But then you still do this and you send your grants in and they still complain. And why do they complain? They complain because there are people that believe that if you're not in a mouse, you're not a biologist, okay, if you're studying pathogenesis. And so, but I sort of always make this argument that if we go back to our black bear, yeah, maybe we're not perfect, right? But at least the bear is outside now, right, I would say. It's breathing oxygen, it can interact with things, it may be sitting on a hammock, right? And I'm not saying this is like being in a human, but it's a lot closer than LP was. So what do? Now you go back to animal models, right? And this is when everyone thinks that you're doing really well. Well, I'm here to tell you mice are not humans. I don't know if you know this, but mice are not humans. Primates are not humans, right? Mice are particularly not humans, okay? But a lot of microbiology has been if you put into a mouse model, you're golden if you see a phenotype. Okay? Now, in Pseudomonas, there's other things other than mice to actually use. There's insect models, there's Drosophila, there's C. elegans. A lot of these models have been used for virions. Okay? If you do mouse models, you think the insect models suck and you say they're not useful. And if you work on the insect models, you say you can do more replicates than the mouse people. Okay? If you read grants, you can just skip the intro because I just told you what it'll be. Okay? That's why they do that. All right. So we've actually done a lot of this. The nice thing about this, right, that allows you is this top one. This allows you to look at interactions with the host to do immunology, right? That's the nice thing about the mice, right? So we've used mice for a lot of different things. I'm going to show you only one set of experiments. This is, again, just 2 slides. And I just want to show you this because I think some of you might be interested in it. So every other piece of data I'm going to show you is on Pseudomonas. This is on Staph aureus. And this is a paper from a postdoc in my lab. And we were asking a very simple question in this. Again, she was using Tn-seq, so this ability to look for genes that you need to grow in an environment. And she actually just took Staphylococcus aureus and she put it into a mouse and said, what genes does staph need to grow in a mouse? Right? What does it need to be able to do? Well, that's not really that interesting. I mean, it's kind of interesting, but not really. But then the next question is, well, how do other bacteria actually impact this? We know we never see staph by itself. A staph monoculture infection is virtually impossible to see. You'll see it a couple of times if you see like flesh-eating things, these flesh-eating diseases, almost always there's another bacterium there. Even if you don't grow it, there's more bacteria there. They're often anaerobes. But we know Pseudomonas is there. Does Pseudomonas impact what staph needs to actually cause an infection? And so it was a really simple experiment. So she did Tn-seq with staph by itself and with Pseudomonas. And you're asking this question, how does one impact the other? And she came up with, and what she found I think was really surprising. And that is, when you have another bacterium there, the things that staph needs to survive and cause an infection completely change. The genes it needs, the functions it needs, the tasks it needs to accomplish completely change. I want you to look at 2 numbers, and then I want to tell you why I think they're interesting. What you're looking at are Venn diagrams. This is a Staph aureus growing by itself in an abscess model of mouse infection, osteomyelitis model of mouse infection, and a chronic mouse infection. These are all staph by itself. This is the one when staph is co-infected with Pseudomonas. Now I want you to look at 2 numbers. This number here, 182. When you put Pseudomonas in, there are 182 new genes staph requires to grow in a mouse and cause an infection. So why is that important? Because it could be that there's 182 new drug targets, right, that people who are doing monoculture infections are not looking at, because these are required, but only in the context of communities. The more interesting thing for some of you is this number here, which is 10. These are genes that staph only needs when it's by itself. But when you put Pseudomonas in, it doesn't need them anymore. Why does it matter? It matters because Novartis, when I went there, was actually developing a drug to inhibit one of these genes. And it was pretty far down the pipeline. And so you're developing a drug that only will inhibit a function that's required when staph is by itself, but it's never by itself, right? And so trying to understand community interactions is key, not only to understanding how to develop drugs, but also thinking about more broadly the community. All right, so this is what people would tell you that work on mice. All you got to do is just find a couple of them, give them a beer, and they'll tell you this. It's a natural infection. It's the black bear outside. It's not on a hammock anymore. It's hunting. It's out in nature, right? And this is how people think about it. But the question is that true? This is a really, really hard question. And no one's really developed a method to do it. And I'm going to tell you a way that we're doing it that I think is a way to do it that will allow us to answer this question. And that is the question. All these models that we write pages of justification about, how can you actually evaluate them and ask when they're useful and when they actually mimic a human infection? And the way that we're doing this is doing transcriptomes, which is essentially metatranscriptomics, just means doing it from a lot of different bacteria. But it's transcriptomics, which is sequencing the RNA from bacteria and using that RNA to infer what functions they're doing. And we'll get into that in a second. So this project was actually from two people in my lab. One's a grad student, Justine, and then Dan Cornforth. One of them is a math person, the other one is a microbiologist. You can tell the difference. It's pretty easy. And I've been trying to do this project since 2003, essentially, and the technology hasn't been robust enough until now for us to actually do this. And the main problem is that we wanted to actually do transcriptomics from human samples. We wanted to take a human sample and ask what genes are Pseudomonas expressing in a human infection. And that's been really hard. There's been a really concerted effort by at least 10 labs trying to do this since the early 2000s. And we finally got it to work. And there's a lot of tricks to it, but we can get it to work now. And so, the data I'm going to tell you about today are, we took a sample, expectorated sputum sample from 7 CF, individuals with CF, 6 people with chronic wounds, these are debridements that people are taking out for us, and then burn wounds, which were also, these were punch biopsies from burn wounds. Okay, and did transcriptomes. And then you're saying, why the hell are you doing this? I'm going to tell you why in a second. So the other thing that we have when we think about Pseudomonas is we know a lot about what it does in these model systems. We know a lot about in vitro. We know a lot about genes it's expressing when it grows in LP and in a mouse and these kinds of things. So we know a lot about this. Not so much about the mouse. We only have 1,000 mouse transcriptomes. But we know a lot about gene expression of Pseudomonas here and here. But no one had ever been able to do it here. So you're asking, well, how can we actually leverage this to actually do what we want to do? And remember, the question we're wanting to do is, can we come up with a way to ask if model systems are good or not? Okay? That's all we're asking. Are they like humans or are they not? All right. So there's a few details we need to go over. The human samples that we got, they're from different infection sites, right? It's burns, chronic wounds, and CF. All these patients were getting different therapeutic regimens. All of them were getting antibiotics of different types. And there were different other bugs there. Staph was generally there with Pseudomonas, but there were other bugs. Most of you say, why did you do this? Why didn't you try to pick a cohort that was very much like each other? And it's because I didn't want to do that. And because I think that really it really would hurt this sort of idea if it only worked with certain cohorts of patients. The other thing you should know about RNA-seq. So what we're doing is we're taking something out of a human, we're extracting RNA, and we're sequencing it. You get, 250 million reads. Some of those will be bacterial reads, and then you can actually use those reads to infer what genes it's expressing. But the thing about RNA-seq is it's a single time point, right? This is a problem. that we've got it that day. We don't have another sample from that person yet, right? And also, the other thing that we think about is your averaging gene expression. Remember I showed you that sputum at the beginning that was 10 to the 9 cells? All those cells are not expressing genes equivalently, right? So you're taking them all and then averaging, right? Good or bad, that's what we're doing. All right, so the first question that we ask is, can I tell an in vitro gene expression pattern in Pseudomonas from a human, right? Is it different at all? And the answer is yes, I can tell. And we just compared this to this, and what do you see? The one thing that we saw in humans were genes that are important for resistance and tolerance to certain antibiotics were highly expressed in humans compared to in vitro. So when they get into a human, you're actually turning up genes that are letting you tolerate antibiotic treatment, okay? That's the main thing I want you to sort of think about is this. But what you end up when you do this kind of experiment is an Excel list with 5,500 genes and numbers of expression. And that's just not very satisfying. And this doesn't allow us to test our models like we want, right? Remember, our goal is we want to be able to ask, is this model like a human? And if it is, in what ways is it like it, and in what ways is it not? So you need something a little more sophisticated. So we went to some machine learning methods that we don't need to talk about, but basically it's like any kind of email filter. I mean, except it works. This works. So what you do with your email filter, right, is you may get a bunch of emails and you tell your computer which ones are spam and which ones are not spam, right? And then it assesses this and then comes up with a way to tell spam from not spam, right? That's the goal. We're going to do the same thing, but we're not going to use emails. We're going to use gene expression of all the 5,500 genes. And then we're going to ask the question, is there a gene expression pattern, a set of genes that can tell us if we're in a human or not, right? We use something called support vector machines. We can talk about it later, but I'd rather get to this. So this is the question. Using the machine learning, are there genes whose expressions can distinguish these? And if you can actually distinguish a human from all of our models, then we can see how a human differs, right? And then we can compare each model also. And the answer is, obviously, that there are a set of genes that you can use. So these 19 genes, in fact, the top 5 genes are sufficient to do this. So you can give me a random transcriptome. I can look at 5 genes and tell you if it was in a human or not, okay? So this is good, right? So this is telling us there's a signature from growing in a human, right? That's kind of cool, right, compared to our model system. It also tells us that none of our model systems are encapsulating these aspects of humans, right? These are things we're missing about humans. So if you look at these genes, there's a lot of these genes. This is their fold change in a human. What this means is this gene here is expressed 4 times higher in a human compared to in vitro conditions. Okay, that's what that means. You can go through and look at this. So you get really excited when you see this, but then you get bummed. Why am I bummed a little bit? This is the gene name. We don't know what any of them do, right? These are a set of genes who have no known function, right, in these bugs. Yet these are the ones that are the signature for human infection, right? One of the problems we had when we were doing this experiment is I started thinking, you know, one of the things that humans have that most of our in vitro conditions don't have is they're all being treated with antibiotics, right? So the question becomes, right, is this just antibiotics? Right? Is it just because they're all getting antibiotics and that's giving you this signature? Well, luckily my lab has enough data to answer that question because I can tell you most genes that are differentially regulated with antibiotics, and only one of them is. So that makes us feel a little bit better, right? The top 5 genes aren't responsive to antibiotics, because if that would have been, we would have published this paper in a crappy journal and just left, right? Because that was just not really that useful. All right. So now that we have this, we have these genes, right? Now we can start asking the question we wanted to ask, and that is, well, how good are our models? Everyone thinks the mouse models are great, right? So how does the mouse model do? So then what we did is you actually do this machine learning again, and then what we do, after it comes up with the genes or the rules by which it can tell an in vitro from a human, right, you feed it the mouse transcriptome, but don't tell it if it's human or in vitro, and then say, where would you put it? Right? And then you can ask, are you in vitro or are you human, right? We did that for 5 mouse models. So this is a mouse burn model. This is an acute model. This is a chronic tissue infection model on the back, and this is a mouse pneumonia model that's used a lot for CF. So this is the fraction that are called as human. So what we're looking at, this is an acute model. That means that I ran this 100 times, okay, and then asked, how many times are you human? And it was 7 out of 100. So 93 times, this mouse actually classifies as in vitro. So it's more like growing in LB than it is growing in a human. Same thing we saw for the mouse pneumonia model that we had been working with. But the mouse chronic model, that was a tissue infection on the back of the mouse, did a little bit better. About 70–75% of the time, it was clade as humans. All right, so that's pretty cool. I mean, it's cool unless you're using burn wounds to mimic CF infections, or pneumonia models maybe to mimic CF infections. But this is still a little bit unsatisfying, right? Because it's just a number, right? It just tells you like a fraction of the time. But the cool thing about these sort of approaches is now we can actually go in and actually ask, well, why aren't these human? What are the genes that are making them not human, right? And these are the genes that are making this one not human. These are our top genes. This is its fold change in a human. This is its fold change in our chronic model, right, which did pretty well, right? And this is our fold change in the burn model. And so now what we can do is go through and start understanding which genes are causing this. And if we know the functions for these genes, we can actually say, we're not functionally able to recapitulate X in this model, right? So this model is not good for studying gene 2911. The chronic wound model is, but probably not this one. Right? So you can begin to ask and find the genes that are important. This, I think, is a little more satisfying. But you also look, there are certain genes that are really good, that are turned up in all the models. But then there are some when both fail, right? But by doing this iteratively, now you can actually go through, if you have enough human transcriptomes, to understand what model would be best for studying a diabetic CF patient, right? Which one would be best for studying a burn wound, right? And we can ask the questions now in a sort of more quantitative way. All right, I want to show you this experiment. It's not published, but I think it's cool. The reviewer did not think it was cool recently, but I think it's really cool. So you guys got to give me some validation. So what we did is there would be times, right? One of the things that I think is real critical that you need to know about bacteria is bacterial RNA is not very stable. The half-life is about 3 minutes. Okay, so all turns over in about 3 minutes. So when you do RNA-seq and you get a human sample, you have to do it very quickly. And if you don't, you're basically not studying it. What most people say, you're not really studying what's in the human. So, but I said, well, nobody's really ever asked that question. We know that the half-lives are 3 minutes, but maybe when you remove a sample from a human, maybe they still have a human-like physiology for a little while, right? Maybe we have longer than 3 minutes. So we asked this question with wound samples, and we took debridements from 5 individuals, and we actually incubated them on a bench top for 6 hours. We just took them out like someone would have taken it in the clinic, set it on the bench for 6 hours. Then we extracted RNA. And then asking the question, would you still be like the ones that we immediately extracted RNA from, or have you changed, right? So the goal would be, when I'm asking someone to take these samples, and particularly these debridement samples are sometimes really hard to get into RNA protection agent, do they have a little bit more time than we think they have, right? And so what we did is we just stabilized the RNA, did RNA-seq, put them in our SVM model, and said, are they like humans, or are they not like humans, just like we did the mouse? And what you see for these samples is 4 of them are really like humans. So 90 out of 100 times they played as humans. This one didn't do so well. And again, we're sort of expanding this study now with the hopes of trying to convince people. My lab has been trying to develop assays, antimicrobial resistance assays, using human samples. But people always say those will never work because as soon as you remove it out of the human, they're no longer human anymore. They do this, and these are sort of things that are thought because of intuition but not science. And so I think we have a method where we can test this now, right, and then ask. And so the goal is we're doing different incubation times, right, out on the benchtop and asking how much time do we have, right, to actually do something where we'd be treating something that is like a human. Okay? So So we're doing a lot of different things now. We actually have just gotten 50 samples from Arlene from Emory, and we've processed most of those for RNA-seq now, expectorated sputum samples, and so those are going in. So that's going to give us by far the biggest data set in the world. There's only one other lab, and they only have one transcriptome. And so we're doing a lot more samples. We're doing every model in Pseudomonas. We're doing other bugs other than Pseudomonas. The other thing that we did, which was a really cool set of experiments, I just wanted to tell you, I don't have the data yet because we're still processing. Yeah.
Can I, sorry to interrupt you,
No, go ahead.
ask you a quick question. You said you incubated it for 6 hours.
Yes.
The typical culture of a bacterial specimen in our laboratory is anywhere between 24 and 48 or 72 hours.
That's right. Yeah.
Do you know how that bacterial profile changes in that longer?
If you take them and put them on a Petri plate, it'll change in 5 minutes. So I think what's keeping them similar now is they're staying in the same context they were in the human. Because, you know, when you debride, you just take tissue out and you sit it aside. They're still spatially organized, right? They're still roughly in the same nutritional environment, probably, because it's tissue. The only thing that's probably changing is oxygen getting in, because now you've given more surface area. And temperature, obviously, temperature is going to be changing. But yeah, if you were to take those bugs out and put them on a Petri plate, within minutes, they would change their transcription. Yeah. So the other thing that we did, and we don't have the data yet for, but I thought I would tell you about really quick, is we actually are looking at biogeographical impact. And this is this biogeography, this idea of where things are in space and time. We actually took a human wound sample and we subsampled it. We want to do the same thing with CF, and we subsampled it. And then we started asking, are the bacterial functions you need to grow here? So when Pseudomonas is here, does it do a different set of things than it needs when it's in the middle, right? And that's almost exclusive. It's going to be true that they do, but we need to sort of begin to define those kinds of things. Because the real question is, yeah, you treat this wound, and then you take a big piece of it, and you go, oh, there's still 10 to the 6 bacteria. But maybe only the 10 to the 6 are only on the outside, right? We don't know where they are. We don't know where they're surviving. And this is something that we really need to think about. Because heterogeneity, particularly for antibiotic accessibility, is probably one of the biggest problems that you're facing, is that the physiologies are so different in the bacteria that are in that infection. And you're trying to hit them with one thing, right? And susceptibility is going to be a little bit different. Anybody know what time it is? Okay, cool. So this is kind of where I was going to stop and sort of give you some sort of hopefully gave you a little bit of an idea about biogeography and the things that we've been doing. And yeah, if there's any questions. Yes, Arlene?
That was outstanding. I have a question and a comment.
Yep.
First question, you glossed over your PNAS paper about decreased expression of core-7
Yeah, for sure,
molecules. Yeah. So does that mean that so that the CF sputum, I guess, as soon as when it comes out of the patient, really doesn't have a lot of core-7 molecules compared to the test tube? So is that where you're thinking that really biofilm may not be so important?
Yeah, I mean, I've never believed that the biofilms, as told to be biofilms, are important because they're not there. You know, these mushroom shapes, they're not there. I mean, I put a couple of slides after this. I wanted to show you, I can show you an image of, in case you guys wanted to see any of the expanded, what is that? Oh, here it is. This is an image of CF sputum. A confocal, a 3D image of CF sputum. The blue is host material and the red are bacteria. I had to reduce the resolution because the file was so big. But that's what a CF sputum looks like. We've now done 17 expectorated sputum samples, and this is what it looks like. It looks like exactly what we kind of thought about. It's mostly host material. Bacteria aren't. A lot of the volume is not bacteria. But if you actually look and you ask, well, what are the structures? They actually are growing in these little clusters of bacteria, and you can quantify those clusters. This is actually, these are the biggest ones we can find in any of our samples, kind of what they look like. So when you're treating a CF patient, this is what you're treating, right? You're treating sputum, at least from an expectorated standpoint. Who knows what it looks like, the stuff they can't expectorate, right? It could be different, right? We don't know that. I suspect it's not. Some people would tell you it is. I don't think it would be. because we see this also in wounds in mice. Everywhere they grow like this. And so if you quantify these clusters, what you actually see, that's the quantification. So what you're looking at is the amount of, the biomass means the total amount of bacteria there. You're asking what percentage are each of these buckets, and these buckets are size of the groups that you see. This one is less than a group of 100 cells. In fact, most of what we see sputum is are cells of 2, 3, 4, 5 in a cluster. Generally, these little groups that are sort of spaced out. And if you ask, is the spacing random? It's not random. They know where the others are. They are spacing themselves out in very proper groups. And so what we just did was an experiment in CF where we took an expectorated sample from an individual, from 4 individuals, and then we gave them, then they took tobramycin. And then 6 hours later, we took another sample. And what we're asking is who's dying? Are you killing the little ones? Are you killing the big ones? Is it just random? Is it stochastic? So, or is there a certain size, right? Who are you targeting with the therapies you're using? Because by knowing that, you'll know who's tolerating the antibiotics. But in regards to quorum sensing, I think for sure, basically the data is that the in vitro systems really overestimate quorum sensing for what you see in human infections. They're still making signals, and they're probably still quorum sensing, but not to the same levels that you see.
Are those clusters cloned?
Yeah, so that's a really good question, right? I usually, we know that when we do this in our synthetic sputum, in vitro, they're clonal, for sure. But it's hard, you know, when you get out of the human, I think we can do it, though. And we've been thinking about ways to do this. And so that's the next question that I have someone, I just have a guy coming into the lab and postdoc and asking one of the questions.
And the follow-up on that is, what's the rate of genomic mutation in these bacteria?
Yeah.
And how much of it is changes in transcription, and how much of it is, random mutation and selection?
Yeah. So mutation rates are actually known primarily in vitro, and this is all we really know. And E. coli can go through, if you go from E. coli from 1 cell to 1,000 cells, there'll be 1 nucleotide change in E. coli in one of the bugs, right? It'll happen one event. So here, it's really hard to know because you don't know how many events they've gone through. But also the environment dictates this mutational rate, right? And so we probably have a much more mutational rate, here. What we do know is if you sequence a bunch of bugs from a CF individual, they are very different. Like, it's often really hard to actually find 2 that have the same genome, right? So they're actually quite different. But what we're doing, but your question of clonality is actually a really good one. And so, because we'd like to pair that, the grant I'm writing is to pair that on top of this antibiotic experiment and ask, is a certain clone living, right? Or is it a certain size? These are sort of the questions. It's kind of amazing we don't know this, right? I mean, you're just giving everyone all of this stuff, and no one even knows what it looks like.
They're hard things.
Yeah, because they're hard things. Yeah.
What is the most impactful, mechanism for the bacteria to be growing, or like a gene mutation, like for the more nutrition, like a nutrient, or like a rheology type, or the mechanical force, or the oxygen content, or what it is?
All of it.
All of it.
But what dictates the physiology of microbes more than anything else are the nutrients that are available almost exclusively. So a bug like Pseudomonas can grow on 110 carbon sources, okay? But it acts differently depending on which one is actually there. And it will actually change how virulent it is. It will change its antibiotic susceptibility based on that. That's really important. Oxygen for Pseudomonas is another one that's really important. Pseudomonas is very odd in that it can't ferment, or it can't ferment in a way that allows it to grow. Things like staph and strep and all of them, they can grow with without oxygen. They don't care. Pseudomonas needs oxygen or nitrate. There's not a lot of nitrate in the CF lung, and so it needs oxygen, and oxygen dictates a lot of its phenotype. Pseudomonas actually swims toward oxygen, toward high concentrations of oxygen. And so I would say those are 2. The physical forces, there are some mechanical forces, but those are usually only really important on a really stiff surface. When you hit a stiff surface, we know that Pseudomonas, when it hits a stiff surface, it changes its physiology because it can tell the stiffness. But an epithelial cell layer is not going to have that stiffness, so I wouldn't think that would be as important, just of the ones you mentioned. Yep.
So I just had some questions about when you were looking at how well your mouse models mimic a human infection, and you looked at expression profiles.
Yep.
So you started with pooled data from expression profiles from different sites, right? Chronic wound
Yeah,
infection.
we started with just human. Everything we had that we could get that worked in a human.
But it was from all different sites.
Absolutely.
And then when you looked at the mouse models, what you found was that expression was different based on location, right? So your chronic wounds kind of had a different expression.
So the burn wound and the chronic wound are the exact same spot on the mouse. One's acute and one's chronic.
Oh, yeah, but they were different infections, right? And they had
They're
different.
infecting the same site, but the outcome of the infection is the same.
Yes. So, but in your human model, you had sort of pooled those things. So have you gone back to look at that pooled data to see if there are differences in expression in those
Oh,
subsets?
yeah, I mean, obviously, that's the first thing you do, right? And so we have done it. The problem is that generally you need more than the number of transcriptomes we had to actually do that. But now we have 50 from Arlene for CF. We know what's going on in a human CF patient now pretty well, right? I mean, 57 is enough for us from multiple clinics. Because I have a clinic in Denmark I work with also, just completely distinct sort of therapeutic regimens, ways they treat are very different. And we see really high concordance in Pseudomonas transcription between the centers. And so that makes me feel a little bit better. But the reason we haven't sort of done exactly what you're saying is we needed more transcriptomes. But we've been generating. But you're right, I mean, that's what you want to do, right? I mean, we just wrote a grant to actually take the sputum samples from humans and say, well, is a diabetic CF patient different from this? And use the machine learning to ask that, right? Maybe the physiology, right? Different therapeutic regimens. You know, all of these things can now be done, but it requires a lot more transcriptome. And it's really expensive. I mean, that was $50,000 for those things, probably, and maybe a little more. Yep.
I guess the final question, to go back to Dr. Chang's question. Your answer suggests that our antibiotic resistance testing may be fatally flawed.
Well, yeah, I mean,
I wonder if you could address that.
yeah, so I think your antibiotic resistance testing will be useful if you have genetic determinants that provide high levels of resistance. If you acquire a gene that produces a beta-lactamase, you're going to be beta-lactam resistant, right? And you don't need new media to test that, right? But the evolution of tolerance in situ, right, is going to be environmentally contextual, right? Where you evolved it, right, that fitness benefit is not going to be a fitness benefit in another environment. And we've done a lot of MIC testing of clinical isolates in our synthetic medium, and then, by the way, the clinical labs do it, and we get completely different results. So sometimes the bugs become much more sensitive in our media, and then sometimes they become much more resistant. And so one of the things that we've been talking to people about doing is actually side-by-siding our synthetic medium with the normal testing and ask whether we're better at predicting. I mean, we don't know. We know we're different, but we don't know if we're better. So I think that's something that we definitely wanted to do. One last thing.
I was just curious about what the genes were that you saw within those 182 genes that seem to be turned on when staph is
With Pseudomonas. Yeah, a lot of them are stress genes. So what you see is Pseudomonas actually puts nutritional stresses on staph when they're in an infection together. So they're competing for nutrients, so it's putting a nutritional stress on there. There's actually some, and that's mainly what we saw were nutritional type, were stresses. So it was a more stressful environment. So the host was not really that stressful. You put Pseudomonas in, it becomes a little bit more stressful. That's mainly what we saw, but there's a lot of different functions.
Well, one more quick question. Of course, all of this work is just fascinating. It's amazing. Pseudomonas and cystic fibrosis have a relationship that is so unique. It's hard to replicate that kind of microbiome disease relationship in any other human disease. From the first infection that a young infant acquires, Pseudomonas infection, to multiple subsequent infections, do we know how that relationship is changing?
Yeah, I don't think we know. I mean, I don't think we know why Pseudomonas just in the first place. Why not the hundreds of other bacteria that are actually in the soil that cause all kinds of opportunistic infections? I mean, I wrote a paper 10 years ago that no one would publish, that an opinion article that said that the reason for Pseudomonas was because the environment that it actually does exist in currently out in nature nutritionally really do mimic the sputum that you see in lungs. And we went out and went to the rhizosphere and other places and just said this bug's incredibly fit in these sort of amino acid concentrations with these sort of polymers. And that evolution has just made it happen to be fit. It's not anything specialized on that initial infection.
And the quality of the alveolar surface lining fluid with the mucus in it.
That's right. And so no one believes that, and well, no one would publish it. And I think it's right. We didn't have the data at that time; it was just an opinion. But I think we're actually getting data now from multiple environments and strains from multiple environments. We actually took a strain from an oil vat down in Texas that was growing in an oil vat and degrading the oil and eating it. It's causing problems for the oil industry because it was chewing up all the oil. We took it and put it in a mouse, hypervirulent, just as virulent as all of our clinical strains are. And so it has clearly not been in a human in a very long time because it's been in an oil vat. But it still has all of the sort of things it needs just to be competitive in those environments. And I really think that it's that evolution out in nature that's provided it with the toolkit to be effective.
All right.